Good day, students. Today we will have our session number 13, and it will be about virology and the study of viruses' structure, life cycle, classification, and medical importance of viruses. So, virology is the study or the science that deals with the study of viruses. Viruses اللي هي الفيروسات. As causative agent هم طبعا causative agent for many diseases. They lead to many diseases in human, in animals, in plants and any other organisms, even in insects في الحشرات وفي البكتيريا نفسها. ممكن الفيروسز تصيب البكتيريا، لكن كل فيروس عنده specific host. الهوست اللي هو المستقبل للفيروس، يعني ممكن يكون إنسان أو خلايا معينة في جسم الإنسان أو بلانت وجزء معين في البلانت، الروتس، الستم، البرانشز. And even there are specific viruses that infect insects. So viruses, they live within the cell of other organisms. They're human, they're plant, they're bacteria. That's why they infect these cells and cause diseases. The study of these viruses, knowing their structure, knowing their life cycle, knowing their classification, their medical importance, we call this study as virology. So viruses, الفيروسات, these are smallest. They are the most smallest and simplest form of life on earth. They very small compared to bacteria, compared to fungi. They are very small. Their diameter usually does not increase more than 20 to 500 nanometers. So they are very small. So viruses, they replicate or they divide or they increase in number within the cells of the host. The host, as we mentioned before, that these cells that can accept the infection of the viruses, because viruses are very specific in their cells. For example, when we say, for example, the COVID virus. The COVID virus is a virus that is specific only for the respiratory cells. So it infects only the respiratory cells. There are other viruses which only infect, for example, the intestine. There are other viruses which only infect the skin. So they are very specific in the cells they infect. This cell, we call it the host for this virus. So virus's structure consists of three main parts. Nucleic acid, that is, it contains a nucleic acid that is either DNA or RNA. It doesn't contain both, either DNA or RNA. It consists of a protein coat that surrounds this nucleic acid, and this protein coat, we call it capsid, as you can see here in the figure on the section note. Some viruses, they have an envelope. An envelope, it is another lipid membrane that surrounds the capsid from outside. So this envelope, we call it envelope, is not present in all types of viruses. It's present on certain type of viruses. But again, it is considered as a character or a morphological character of this group of viruses that have envelope. The infectious particle of these viruses is called a virion. So when the virion, which is a single, when this virion infects a host cell, we call that this cell is infected by a virion. The virion, it is a completely infectious virus particle. Either it is DNA, contains DNA or RNA, or it has an envelope or doesn't have an envelope. So at the end, any type of virus that infected a cell, we call it a virion. So what is the general characteristics of viruses? As I mentioned before, that the viruses particles are the smallest and simplest because they don't have organelles like in the bacteria or in the or in the fungi. They have a cytoplasm, they have a nucleus, they have organelles, but viruses, they don't have all these organelles, or they don't have any cytoplasm, only nucleic acid and a capsid protein, and some viruses have an envelope, which is a lipid membrane. But the most important characteristic of the viruses, they are very small, and their size is between 20 to 500 nanometer. Unlike bacteria, its size approaches micrometer, which is bigger than the viruses. So viruses are called obligatory intracellular microorganisms. That means they only replicate. Obligatory, that means it's mandatory. They have to replicate within the cell. They they cannot replicate outside the cell. They use the organelles, they use the enzymes of the host cell to replicate. That's why we call them obligatory intracellular microorganisms. They multiply, as I mentioned, within the cell, the host cell, using their organelles, using the enzymes, using the ribosomes, using the energy that is found within these host cells to replicate itself. That's why the virus infection is usually very difficult to get rid of it, because it replicates within the host cell. It usually manipulates or uses all the facilities and the organelles within the cell for itself to replicate. So that's why the cell gets ill and most of the time dies. The other characteristic of viruses is that they do not contain any organelles. That's why they are very simple. They don't contain ribosomes, they don't contain tRNA, they don't contain any metabolic enzymes, but they use those enzymes within the host cell for itself. Viruses are not affected by antibiotics. Unlike bacteria, bacteria can be killed by antibiotics, but viruses cannot, because they replicate within the host cell. If you want to kill the virus, then you will kill the host cell. That's why it's very difficult to treat a viral infection. But there is a known drug that is used now in most infected people, which is the interferon. Interferon, it does not kill the virus, but it stops its replication within the cell. So it interferes. That's why we call it interferon. Interferon, because it interferes with the replication cycle within the host cell. So it stops its replication and its increase in number. So interferon is one of the drugs here used in viral infections. Viruses, you cannot grow viruses on artificial media in the laboratory, but you can grow them in what we call tissue culture or cell culture media that contains cells, because they need living cells to replicate. There are specific type of cells or tissue culture cells or tissues. They grow, it's used in the lab to grow these viruses and to study them within the lab. Some viruses, they cause latent infection. When we say latent infection, that means they infect the host cell, but they do not cause symptoms of disease. They stay within the cell and became dormant or latent infection, that is doesn't show any symptoms, but this at the end the cell is infected, and this happens in large number of infection in human beings, that the virus it is replicating within the body of the human being, but is not causing any symptoms until it reaches a certain point. Then very severe symptoms appear, and this type of infection we call it latent infection, اللي هي العدوى الخفية أو latent أو dormant. Viruses cannot be seen by ordinary. You cannot see virus in the laboratory by using light microscope. We only see viruses, or if you want to study viruses, you use electron microscope. So this is are the shared characteristic of the viruses that infect human, animals, plants, bacteria, and other type of living cells. We are going to speak next about the structure of viral nucleic acid. As for the structure of viral nucleic acids, this is the genetic material of the virus. So it is found, one of these four types is found in the virion, which is the infectious particle of the virus. So the virus contains one of the four types of structures of the nucleic acids. It's either double stranded DNA or single stranded, that means one strand of the DNA, or single stranded RNA, or double stranded RNA. So the virus usually contains one of these. No virus contains more than one type of nucleic acids. So that's why these nucleic acids are the content of the virus for a specific nucleic acid is used as a classification. We are going to speak later about this as the classification method for the viruses. So the type of nucleic acids is either double stranded DNA or RNA, or single stranded DNA or RNA. So how can we differentiate? How do scientists differentiate between this is a DNA virus or an RNA virus, regardless if it is single stranded or double stranded? To differentiate if it is DNA or RNA viruses, they use the enzymes that is specific to degrade this, to hydrolyze or degrade or lyse this nucleic acid. If the virus lyses with the addition of DNase enzymes, then this is a DNA virus. If it lyses with RNase enzymes, then this is an RNA viruses. So there are specific tests to test if this is a DNA virus or RNA virus. To test if the virus is single-stranded or double-stranded, then they use certain type of dyes or stains to stain the infected cells. So with the virus, so they use you can use acridine orange stain, which is yellowish green. If the which turns to yellowish green if the virus contains a double stranded, and and turns to red orange if the virus is single stranded. So you can differentiate if it's DNA or RNA with the enzymes, and if it is single or double stranded with the use of a acridine orange stain to stain the infected cells. And if according to the color that turns the this stain, then you can differentiate if it is double stranded or single stranded. When you speak about viral proteins, as we mentioned, the virus particle consists of nucleic acid surrounded by a protein coat. This protein coat, we call it a capsid. The units of proteins that constitute this capsid, we call it a capsomer. So a building unit for the capsid is one unit of a protein that builds this capsid, we call it a capsomer. So they arrange these capsomers, they arrange themselves to give the outer shape of the capsid or the shape of the virus. They have different shapes. We are going to speak about this later. The virus itself, they have different shape. They are cubical, they are filamentous, they are longitudinal, they are different shapes. So this capsomere, the arrangement of this shape gives the shape of the outer protein coat, which is the capsid of the virus. These capsid or these capsomeres, they are the one that help the virus to attach and penetrate the host cells. So these capsomeres, they help the virus to attach itself to the host cell, and then can penetrate into the cell. That's why these capsomeres, they call it, they have an antigenic, they contain an antigenic determinants. That means they have antigens that help them attach to specific cells. As I mentioned before, viruses, they have specific host cells, so that's why it has an antigenic determinant. That means it is a specific antigen. That means it has a specific shape to fit in a specific host cell. And usually the part in the host cell where it fits, it's called the receptor. So the virus itself is shaped, this antigenic capsomeres, to the shape of the receptor so that they can fit there and enter into the cell. The viral envelope, as I mentioned before, that some viruses, they have a lipid membrane that is surrounding the capsid from outside. As you can see here in this diagram, this is a nucleic acid, which is the single-stranded RNA, and this is the membrane that is the capsid, okay, or what we call the protein, the capsomeres that constitute the capsid, and then this is surrounded by a lipid membrane. A lipid membrane, it's called an envelope, and it is found in in a number of viruses. Not all viruses are enveloped. So those viruses that contain this envelope, we call them enveloped viruses. Those that does not contain an envelope, we call them naked viruses. So the lipid membrane is found in certain types of viruses like orthomyxovirus, paramyxovirus. But this lipid membrane or envelope, sometimes it contains like spikes. Okay, it contains like projections, and these projections are usually made of glycolipids, and they are the one that help the virus, this enveloped viruses, to attach itself to the host cell. So the enveloped viruses, they attach themselves to the host cells through these spikes, or what we call piplomers, to the host cell. While the neck viruses, they use the capsomeres. Okay, they use the capsomeres, which is antigenic determinants, to attach themselves to the host cell. So this is a characteristic of the envelope viruses and the naked viruses. Next, we are going to speak about the replication of viruses, life cycle, or the general life cycle of viruses, because there are certain uh, exceptions, or there are certain alterations in this life cycle for specific viruses, but we are not going to speak about each specific virus. We are going to have the general life cycle of viruses within the cell. As for the life cycle, replication life cycle of the virus within the host cell, the life cycle consists of five stages. The first stage is what we call the attachment of the virus to the host cell. So the attachment of the virus to the host cell is the attachment of the virion, which is the infectious particle of the virus, to a specific receptor on the host cell. So, as I mentioned, when we spoke about the structure of the virus, we said that the capsomeres, they act as antigens that fit to the receptor in the host cell. That's why the viruses are specific to certain receptors within a specific host cell. So they attach themselves to these receptors and these structures within the host cell. And these receptors are where the viruses attach themselves to the uh, outer membrane of the host cell. If it's an animal, if it's a plant, if it is any other type of microorganisms. For example, the influenza viruses use the receptor, what is called the salicylic acid receptor, which is it is a receptor found in the respiratory cells. So that's why the influenza virus infects only the respiratory cell, because these receptors are only found in the respiratory cells. For example, the AIDS, or what we call the human deficiency immunodeficiency virus, attaches itself to certain type of lymphocytes, which is what we call the T cells, the CD4 receptor. The CD4 receptor, it's a type of receptor that is found in the T cells, which is lymphocyte cells, and so on. So, like the rabies virus, it binds to what we call the acetylcholine receptor, and this acetylcholine receptor is only found in neural cells or nervous system cells. That's why the rabies always affects the nervous system cells. So this type of specificity or specific attachment of the virus to specific receptors within specific cells, what we call the process is called as tissue tropism. Tissue tropism is that the viruses are specific in their infection to specific type of cells that contain specific receptors within these cells. So tissue tropism, this is the terminology used for this specificity of the virus. After attachment, then the virus goes to stage two, which is penetration into the host cell. So to penetrate into the host cell, it depends on the type of viruses, but usually it's reported that there are three types of penetration. The virus use one of these methods to penetrate into the cell. It depends on its type. So the first method is what we call translocation. So the virion just crosses from the receptor through the receptor from outside the cell, in crossing the cell membrane into the cytoplasm, and this is what we call translocation. So certain types, they use this method to enter or penetrate the cell. The other type is what we call endocytosis. That means engulfing the cell. It engulf the virus. That means it causes like invagination inside the cell, like a vacuole. Then what we call endocytosis, it engulfs this virus and the virus enter into the cell. Other type of The third type of method of viruses, they use this method, what we call fusion with the plasma membrane. They fuse, the virion particle fuses with the plasma membrane, and once it fuses, then the membrane breaks and enters into the host cell. So the penetration process or the penetration stage, depends on the type of virus, either translocation, endocytosis, or fusion with the plasma membrane. Once the virus enters into the cell, then the third stage happens, what we call uncoating of virus. Uncoating, that means removal of the capsid membrane, uh, capsid, uh, capsid protein, or the protein membrane that covers that covers the nucleic acid of the virus. So this stage what we call uncoating of virus. So uncoating happens once the virus enters into inside the cell and becomes part of the nucleus or the cytoplasm. It depends again on the virus type. Then once inside the cytoplasm, it uncoats the protein coat dissolves, and this lysis of the protein coat of the virus is by the enzymes found within the cell and the change of the pH within the host cell. These all factors results in uncoating, dissolving of the protein coat, which is the capsid of the virus, and leaving the nucleic acid free within the cell. After uncoating, then the viral nucleic acid, if it's a DNA virus, then it migrates and goes to the nucleus of the host cell. If it's an RNA virus, then it remains within the cytoplasm. There are exceptions to this, but we're going to say it later. But this is usually what happens. The DNA virus, they migrate to the nucleus of the host cell, and the naked RNA viruses, the I mean the nucleic acid of the RNA viruses remain into the cytoplasm, preparing for the fourth stage, which is we call gene expression or biosynthesis. So at this stage, the fourth stage of the virus life cycle, then the virus starts to replicate. Viruses do not replicate by binary fission as bacteria, or by budding as fungi, or by mitosis and meiosis. No, they replicate by using the enzymes, the organelles within the host cell for itself. That's why it uses the energy of the host cell. It uses the ribosomes of the host cell. It uses the replication enzymes within the host cell to replicate itself. That's why, as this stage, the infected cell, the host cell, becomes ill. It does not uses its own machinery to grow up. No, the virus uses its own enzymes and energy and organelles for itself to replicate and start what we call this stage of biosynthesis or gene expression. At this stage, the viruses uses all these enzymes and energy within the cell to express its genes and produce proteins for its coat. So the process, this at this stage, there are three processes that the virus undertake within the host cell. The first thing is the replication of the viral genome. So if it is a DNA or RNA virus, it starts to replicate, increase the number of its genome. If it's a DNA virus, then it replicates within the nucleus, except one type of DNA virus, which is the pox virus. It replicates within the cytoplasm, although it's a DNA. So it's something specific to this virus. RNA viruses usually replicate within the cytoplasm, except retroviruses like influenza virus. They replicate in the nucleus, although they are RNA viruses. But as a rule, DNA viruses increase or replicate their genome within the nucleus, and RNA viruses replicate their genome within the cytoplasm of the host cell. So this is the first step in biosynthesis of the genome of the gene expression of the virus. The second step is what we call transcription. And as you know from the biochemistry, there is replication, there is transcription, and then translation. So transcription is the transcribing the replicated nucleic acid into messenger RNA. If it's a DNA or RNA, then it is transcribed or transferred by certain enzymes into what we call messenger RNA, because to go to the third step, which is translation, which is production of proteins, then this is happens within the ribosomes of the host cell. So for the host cell ribosomes to try to start translating the genome of the virus, the message should be in the form of messenger RNA. That's why before translation there is transcription process, and then the translation where this messenger RNA of the virus start expressing the capsomeres and the capsid of the the capsomeres that is specific for the capsid of the virus. And that's why we call this state biosynthesis. That means synthesis of the protein of the virus components, either genome or proteins. The fifth stage we call it the assembly of the virus. So the assembly of the virus is where the nucleic acid is start surrounded by proteins, which is the capsomeres that were translated in the biosynthesis stage. Then they start to assemble themselves in the shape of the virus to constitute the capsid. Once the capsid shape takes place and it is surrounding a nucleic acid that is specific for this virus that is already replicated within the host cell. So this is what we call the assembly of the virus. Once it takes its shape, the virions themselves within the host cell takes their shape after the biosynthesis and assembly stage. Then it goes to the last stage of its life cycle, which it releases itself from the host cell so that it can infect other cells. So viruses releases themselves from the host cell either by either of the two methods. Either they kill the cell, they lyse the cell, and then they free themselves and go to other cells and start new life cycle, or by budding without lysing. Budding is just like the what happens in the in the fungi. Budding in this case it is opposite to endocytosis, it is exocytosis, so it moves itself and pushes itself to the cell membrane. If it is in the cytoplasm or the nuclear membrane, it pushes itself until it moves out of the cell and cuts part of the cell membrane or the nuclear membrane of the host cell. And this method of budding, it happens in the enveloped viruses because the envelope is usually part of the cell membrane or the nuclear membrane of the host cell. That's why they become enveloped, and it is lipid lipoprotein in nature, as the same nature of the cell membrane of eukaryotic cells. So this is the life cycle of the viruses. It has 6 stages, as we described. Next, we are going to speak about the classification methods. There are different classification methods of viruses, and we are going to explain it in more details. As for the virus classification, there are 3 ways of classifying the viruses. The first way is based on the genomic nucleic acid type that is found within the capsid of the virus. So there are DNA viruses and there are RNA viruses. DNA viruses can be single stranded DNA, as we mentioned before, and this is an example for parvoviruses or picornaviruses. It could be double stranded DNA virus, and this is like adenovirus, herpes virus. So these are all DNA viruses with the difference in the number of strands, either double or single. The other type of viruses is the RNA viruses, and this also has 2 types: double stranded or single stranded. Double stranded RNA viruses, examples the reoviruses, and the single stranded RNA viruses, again, This is again divided into two, into positive stranded RNA viruses or negative stranded or negative sense RNA viruses. And this depends, this classification, it depends on the polarity. This is something beyond this session, but I think you already have it in the biochemistry. It depends on the polarity of the RNA strand. So it is either positive or the negative sense RNA viruses. So this type of single-stranded RNA example is the hepatitis virus, which is very well known, which is hepatitis A. It infects the humans. The other type of classification is based on the nucleocapsid shape structure. The nucleic acid, as you know, as we mentioned before about the structure of the virion that consists of a nucleic acid, then surrounded by capsomeres, which join together to give the protein coat, which is called capsid. So this particle of the virus, we call it nucleocapsid. Nucleic, that means contains nucleic acid, and capsid, that means it contains a protein coat. That's why it's called nucleic acid. So, one of the classification methods is based on the structure of this nucleic acid or the shape of this nucleic acid, nucleic capsid. So, they are divided into four symmetries. Symmetries, that means tanazur, or when you see the particle of the virus from different sides, is it there is similarity between from all sides. That's why we call it symmetry. So according to this, they are divided. The classification is based on four symmetry structures: helical symmetry, cuboidal symmetry, and binal symmetry, and lastly complex symmetry. Helical symmetry, this is when the capsid, its shape is like spiral or helical structure. And this is an example of tobacco mosaic virus, which affect the tobacco leaves, and the rabies virus, which affect the humans and cause rabies. So this type of helical symmetry, that means the virus structure or the nucleic acid is in the form of spiral or helical shape. For example, for the rabies, we call it bullet shape. That means it is very sharp helical. These viruses could be enveloped or could be naked. For example, for an enveloped helical virus, example the rinderpest. Rinderpest, it's a type of virus that infects animals, especially cows, اللي هو بيعمل ما يعرف بطاعون البقر. Rinderpest. It's a type of virus, and its shape is helical, but it is enveloped. Um, Other type of virus which is unenveloped, like the tobacco mosaic virus, it is helical but is naked virus. So for the rabies, it has spikes on the envelope, the lipoprotein. It is enveloped, but it has spikes, and these spikes, as we mentioned before, it's an antigenic determinants that help the virus to attach itself to a receptor. The other symmetrical structure is the cuboidal symmetry, and this in this shape it has uh, triangular shapes, which are what we call 20 triangular shape fixed together, what we call icosahedrons, or triangular faces, 20 equilateral triangular faces. And this was studied by the electron microscope using different staining techniques to study this symmetry, cuboidal symmetry. And again, this is usually many RNA and DNA viruses are cuboidal, and they have these 20 equilateral triangular faces. They are joined together by divalent bonds. and it gives its shape the cuboidal shape. They could be enveloped or naked, but they share the type of cap smears and the 20 equilateral faces, triangular faces. An example of this is the adenoviruses. Binary symmetry, from its name, that means it's a virus that combines the two previous symmetry, helical and cuboidal. And this is very common in what we call the bacteriophage virus. This bacteriophage virus, it has a cuboidal head and helical tail. And this type of virus is very known for the infection of bacteria, all types of bacteria that can be infected with this virus. They have this type of bacteria, this type of virus, what we call bacteriophage virus. The fourth symmetry, which is a complex symmetry, and this is neither cuboidal nor helical. So it is a different shape. For example, the pox virus, it's brick shape. That means it looks like a block. And this again, it's different from the cuboidal and the helical. So we call it. It has different other shapes, and this type, when it is classified, we call it complex symmetry. Uh, from these types of cuboidal, helical, and complex, the viruses could be naked and it could be enveloped. From the table here, I'm just giving examples for the naked icosahedral viruses. For example, the hepatitis A virus, the envelope viruses. There are other types of viruses which are also icosahedral, and yet they are enveloped. So this table will show you the difference between the different symmetries, but if they are naked or they are enveloped. We just need to know this information, that they could be naked or they could be enveloped. We are going to move to the third type of classification, and this is the most recent type of classification used nowadays among scientists or virologists to classify viruses. As for the Baltimore classification, this is a recent type of classification of viruses using molecular biology techniques, because this is depends on studying how the virus express in the expression or biosynthesis stage, how it transcribes its messenger RNA, producing messenger RNA, so that it can be translated into proteins in the host ribosomes. So this classification has been discovered or has been announced by the Nobel Prize scientist, who is David Baltimore, and it is named by its name. He's a biologist who received the Nobel Prize in early 19th century, and this classification was announced in 1970s. So the classification, as I mentioned, it depends on how the virus produces or transcribes its messenger RNA. And to study this, of course, you need to utilize molecular biology methods. So, based on the how these viruses produce their messenger RNA, they are divided into 7 groups. The first group is the double-stranded DNA virus as their genome, and they produce the messenger RNA by transcription in the same way as the host cell. The second group, these are single-stranded DNA as their genome viruses. They convert this single-stranded genome into double-stranded DNA, which is an intermediate stage between producing to produce messenger RNA, and then this double-stranded, it acts as the same as group one, and then it is transcribed as the whole cell into messenger RNA. Of course, these all methods they utilize the enzymes of the host cell. Group 3 viruses, these are double stranded RNA viruses as their genome. And these double stranded RNA viruses, one of these strands will act as messenger RNA. So it is used directly to be translated and replicated, while the other RNA, the other strand, then it is used for replicating and using the machinery of the host cell for replicating. But one of the strands of the double stranded, it is itself becomes a messenger RNA. Group 4, these are viruses which have single stranded RNA, and as mentioned before, there are two types of single stranded depending on the polarity of the single stranded. In this case, we are in this group, we are speaking about positive polarity, that is positive single stranded RNA genome of the virus. This can serve directly as messenger RNA, and itself it can produce intermediate nucleic acid like double stranded RNA, but itself it acts as a messenger RNA to be translated in the ribosomes. The fifth type, which is called viruses that contain single stranded RNA with negative polarity. So in this case, the single stranded RNA is complementing to the messenger RNA. That means it itself it cannot act as messenger RNA. So this group of viruses, they undertake different stages within this process to produce messenger RNA. They replicate themselves as double-stranded RNA in an intermediate stage to produce messenger RNA, and then one of these strands, which becomes positive polarity, then it is used as messenger RNA to be translated in the host ribosomes. The sixth type is what we call two copies of single RNA genomes, and this has to be converted. If like this, we call them these viruses. We call them segmented viruses. That means it is an RNA. It has single stranded RNA genome, but two RNAs with different sequences. So this has to be reverse transcribed into double stranded DNA, and then this double-stranded DNA transported to the nucleus as a DNA molecule and integrate itself within the host genome. This is how this virus gets usually cause what we call cancer cells, because it integrates itself within the genome of the host cell, and then automatically it is transcribed with the help of the enzymes as part of the genome of the host. And that's why these viruses are more dangerous than other types, which is group 6. Group 7, these are viruses which have partial double-stranded DNA genomes. When we say partial, that means um, they don't have all the sequences that is required for DNA to transcribe itself just using the enzymes of the cell. So it produces single-stranded RNA as an intermediate that act as messenger RNA. So they lack the reverse transcriptase enzyme for genome replication. So that's why we call them. They don't carry the whole sequence that help them use the enzymes. Of course, these host enzymes, they need specific sequence within this genomic viral DNA, so that it can attach itself and facilitates the replication of the virus. So that's why we call this group partial DNA. They are DNA, but they do carry the specific sequence that help the enzymes of the host to attach itself and start replicating and transcribing the DNA. That's why they produce single stranded RNA intermediates that act as messenger RNA. The table shows the seven groups as their and just give examples, more examples for the names of the virus for the different group. You just need the most important, you just need to know the groups and the specific character of this group, if it's a single stranded and how it produces its messenger RNA. That's what you need to know. Last part of this session is the medically important viruses that infect human beings. And here I'm giving you two tables: the most important DNA viruses families and the most important RNA virus families. What you need to know and know by heart from these tables: you need to know the family, if it is enveloped or not, and the capsid symmetry, if it is helical, if it is icosahedral, or if it is complex. You need to know the the type of genome, of course, if it is DNA, if it is single stranded or double stranded. That's what you need to know. And have and know the virus name, the specific virus name for these families that infect humans. So you need, as I mentioned, you need to know the family, if it is enveloped or naked, the capsid symmetry, and the type of genome, if it is single stranded or double stranded. You don't need to know if it is linear or segmented, or you don't need to know this. Just know that if it is single stranded or double stranded. And then to know the medically important viruses that is related to this family. Okay, so this applies for DNA viruses and RNA viruses. Good luck, and wish you all the best.